run in here. So we're going into chapter 13, which is applied genetics. So we looked at how DNA replicates, it mutates, all this yada 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 stuff. How do we apply it now? How do we use all this information? How do people use it? Well, selective breeding is really what it kind of boils down to. And so you guys should have this on there. So the first thing we do is we do something called selective breeding. So this is where we are trying to pass on favorable traits. If I have a tomato plant that just gave me so many tomatoes, I'm going to want to use that seed next year. And maybe what I'm going to do is I'm going to take this seed that gives me really like a lot of tomatoes, and maybe I'm going to breed that tomato seed with another tomato plant that makes really hardy tomatoes. They resist bugs quite well. And, logically, if I'm passing on those outcomes, I should now have a tomato plant that produces lots of fruit that's resistant to insects. I have created a hybridization of this. I have selectively bred it to pass on the traits I wanted. So again, this is where all modern agricultural products come from. If you've ever seen what a wild strawberry looks like, what a wild watermelon looks like, they look nothing alike. Bananas as well. Any fruit, vegetable, if it comes out of the ground, if it's grown, has been either selectively bred or genetically modified, which we'll touch upon later. They look nothing like they looked like 200 years ago. Has anyone ever seen it like a chicken from like the 1940s? Yeah, let me load up that picture. So again, these are the same species of animals. I know it's really small. It's a really blurry picture, but you can really tell the size difference, right? The modern chickens are 5 times the size of chickens from 100 years ago. We have selectively bred it so that the ones with bigger breasts, bigger thighs, etc., have the traits we want. We want more meat out of them. So selective breeding is something that happens in all agriculture. All agriculture. Oh, literally the chicken right there. Again, so these, I know it's blurry, but those are some examples of how that's worked. Another example we usually see is dogs. Before, and this isn't even that long ago. This is like 100 years ago. These before pictures were historically not that long ago. Evolution takes millions of years. What we've done is we've selectively bred these animals for certain traits. For bulldogs and pugs, for example, they've bred it so that they have more mashed up faces. For boxers, I think that's a boxer. They had it where the ears are straight up, for example. So they look different, and this is not always good for the animal. In fact, it frequently is not. Because sometimes a lot of that is done through inbreeding as well, which carries consequences. Probably the biggest bastardization of this is the pug. Pugs have really bad health problems. They can't breathe very well. Their face has been so flattened and mushed together, they cannot breathe very well. That's what a pug used to look like. It used to have a normal dog's face, right? And now when we think pug, we think of whatever this thing is, right? So, oh, sorry, I have the wrong thing open on my side. 1 second. Here we go. So hybridization involves cross-pairing organisms with different forms of a trait to produce offspring with specific traits. Like I said, bear with me. I just have a trillion things open. So again, if I have a tomato with disease resistance and I have one with large offspring, ideally I will have large disease resistant tomatoes, right? And I guess for context for me, for those of you here, is that enough space to like write in, or is that too small on your page? Like having the 3 slides like that, is that just not enough space to write in for those blanks? It's good. Okay, yeah. Because I can make future ones bigger if it is a problem. So just let me know if that is the case. But okay, so that's hybridization. Now, is anyone here a Game of Thrones fan? Or is anyone watch Game of Thrones? No, few. Well, one of the whole things of it, like is we look into this process of inbreeding. There's the Targaryens and the Lannisters, right? And they have a saying in the show that anytime one of them has a kid, God flips a coin. That's either going to be a crazy mad kid or it's going to be a normal kid without this insane temper. And so what we see that in that show through inbreeding. Inbreeding can cause problems, but inbreeding is done purposely to keep certain traits, quote unquote, pure. We see it a lot with dogs. We see it a lot with dogs, right? If I want nice, pure golden Labrador puppies, I'm going to breed my dog with someone else's pure Labrador with paperwork to back it up. But that is different than hybridization, though. So hybridization is 2 different traits being blended. So we breed these to be more disease resistant, to create more offspring, and to grow faster. The downside here, though, is it takes a lot of time and it's really expensive to be able to do. Inbreeding, however, is a little bit different. So inbreeding is when we take 2 closely related organisms or that they have a specific trait, and we want to pass on that desirable trait to future generations. So we looked at that transformation of the pug. How did they do that? They took 2 pugs with very short snouts, like, oh, this one has a short snout, this one has a short snout. We breed them together. Their children are going to have slightly shorter snouts, and we repeat the process. So there are, of course, problems with this. Inbreeding can increase the chance of homozygous recessive traits, that homozygous recessive offspring that might have those harmful traits. What does that mean? Homozygous recessive? If, let's say if C, if C is cancer, if I have an animal with this genome, this is homozygous recessive here, right? Now, this animal gets cancer. But if it had something like this, if it was heterozygous, or if it was homozygous dominant, it might be that this gets no cancer, or, substitute cancer for whatever it is. So why is inbreeding so bad? Like, typically speaking, for the health of things, how come every Labrador like dies at 8 years old? Every pure Labrador, any pure dog, how can they have a ton of health problems? Purebred dogs have a ton of health problems. Why? Because they've been inbred to the point. There we go. That they have these homozygous recessive traits. It is much more likely that they get that. So that's why inbreeding is typically taboo. But we do it a lot for animals and plants. So this right here is probably the most famous pharaoh, King Tut. So when we found him, we found his coffin here. They opened it up and they reconstructed what his body was supposed to look like. And this is what we have on the right here. King Tut was a product of incest, of many generations of incest. he had a ton of health problems. I don't fully remember them all off the top of my head, but he didn't have like a working leg, for example. Like his bones were different sizes. He had a ton of different like health problems, for example. So ton of problems. Inbreeding causes problems typically. It causes health problems. We do it for dogs because we like what they look like, but it's not a good thing. So how do you healthfully do this? If I want to pass on a trait, if I want to grow really strong, tall horses, but I can't inbreed them. It's going to cause problems. We know this. So how do I create healthy future generations of tall, strong horses? You add something that's not in the family tree. This is an example here of adding stuff in our family tree. It can eventually create problems. If this one over here, however, gets married into something not from the family, that's going to be better. So when we inbreed, it's again it's made in those closely related individuals to preserve the characteristics, but that does not mean it's healthy to do. So all of a sudden, again, there's a little bit of inbreeding in here, and now there's an offspring here that has this recessive trait, that has this undesirable trait that's emerged from it. Which kind of leads us to this. So we've seen this stuff on the right before, right? Those are our Plunkett squares. So what we do is we take a test cross and we breed an organism that has an unknown genotype with one that's homozygous recessive for the desired trait. So again, I have an unknown genotype. I don't know what it is, what its genetics are. You can tell by phenotypes. You can't tell what genotypes are. So if we have a parent and the unknown genotype is homozygous dominant, so homozygous WW, if it's homozygous dominant, all the offsprings are going to have that dominant feature, right? We know this. But if I have an unknown parent and it is heterozygous, then that means half of the kids are going to have the recessive trait, half of the kids are going to have the dominant trait. So one of the really complicated things of these Plunkett squares is when we don't know the genotype of a parent. You have to use logic then to kind of figure it out. You have to work backwards. This is an example of that. Okay. And that's it for this first part. We just needed the concepts of it. So the main concepts here are selective breeding and inbreeding. What's the difference between them, right? It's just understanding that. And then, of course, hybridization. So there's really just three kind of main things that we're focusing on here today. That said, where's my corn? There's more than one way to increase the size or characteristics of things. namely agriculture. Anyone guess what it is? We go in and modify it. So instead of having to do this expensive process of breeding stuff over 10, 20, 30, 40 years, I can just go into the code and I can do a couple edits and all of a sudden, poof. So that's where we're headed to next year. So we're going to head to the next section, which is section 2. Give me a second just to locate that, especially for those of you online. Like I said, we're moving pretty quick. I apologize, but it kind of has to happen. Okay, so let's get into this here. So we can use genetic engineering to do what we want. We can manipulate DNA. We can read what is DNA. We can read the genome now of things. So if I've identified a gene that, hey, this is the gene that tells you how big a tomato gets, I can just go into it and I can swap out that A for a T or something like that, right? I can go in and change those genes. So that's what we're kind of focused in on here. So what we do is we can take genetic engineering, which is a technology that involves manipulating the DNA of one organism to insert the DNA of another organism. So what we've literally done is we have inserted DNA. So again, literally, I am taking out the DNA and I'm inserting the DNA from a different entity in there. Can anyone think, what's the most famous movie example that does that? Jurassic Park. Jurassic Park, the whole concept of Jurassic Park, for example, was, hey, we have this ancient dinosaur DNA, but it's a little bit broken. So I'm going to take some frog DNA, the missing pieces of frog DNA, and I'm going to put it in there. We're seeing real world examples of this as well. We can theoretically bring back the woolly mammoth. If we were to insert DNA from an elephant into a woolly mammoth, it's viable. They can do it. So Jurassic Park, woolly mammoth are definitely the most common examples of this. So why do we do it? So why are we genetically engineering stuff? One is we can look at the expression of a particular gene. What does it mean, expression? It helps us figure out what a gene does. We can look at cellular processes. And we can look at the development of diseases. So what we can do genetic engineer, so we can use this to increase or decrease the expression of specific genes. So again, what does that mean? Increase or decrease the expression of a specific gene? Again, think about, I found the gene for tomatoes that chooses how big it grows. I can make small tomatoes, or I can try and make as many big tomatoes as possible. So expression is how it's shown. So how do we do this? The big thing we do is we look at their genome. So a genome is all of their DNA. We take it out, we lay it out like a book that we can read it. It's where literally I've uncoded the entire genome. So once I have my genome, I then am going to manipulate it. And I'm going to isolate genes from the rest of it. So if I found the gene that makes my tomatoes smaller than I want, I isolate that gene and I put in something I want there instead. Now, simple in theory, obviously, that's really hard to do. It's not like Lego where I just take out a different piece of Lego. So what I've done is I've included a bunch of tools that we use. We'll go over what most of these are, but these are things that are used in this process. But here's the question. If the whole thing is I'm going to isolate genes from the rest of the genome, how do I do that? DNA is tiny. It's so small. How do I possibly go in and cut it out and replace it? I can't take a scalpel. I have to do something different. So we use something called a restricted enzyme. So what I'm going to do is I'm going to take proteins that are going to bind to the DNA that I want gone. And it's going to cleave the DNA in that sequence. So I'm literally attaching DNA. I'm attaching proteins to the thing that I want gone. And then it cuts it out. Where we get these from was from bacteria and viruses. We found out that these were a defense mechanism. And we use it to isolate specific genes. Yeah. Okay. So we're going to start looking at some of these enzymes. So one of them is we call it echo RI. So this is an enzyme that specifically cuts the DNA sequence GAATTC. Remember, it reads out the genome, and the second it finds that GAATC, which is this stuff right here, it's found this, it's going to chop it out. The reason we chop that out is because now I have a really nice anchor here. I have a really nice anchor to attach another strand to. So these are what's called my sticky ends. These T's and these A's here, these are my sticky ends. This is what I use to connect it. Think about these as those little ridges that rise up on Lego. That's what you connect the Lego to. So these sticky ends are called they're single-stranded DNA sequences. They are at the end of these fragments, right? They're the edges of it that you attach it to. We reattach it then to the complementary strand, to the strand we want. I reattach it to the trait that I want. So cool. So what I have now is I now have fragments. What do I do with these fragments now? So now that I have these fragments here, I'm going to put them through something called a gel electrophoresis. There we go. So I put them through a process called gel electrophoresis. So this is when I literally put in an electric current. I use electricity to separate it. So when I add electricity to it, it turns it into a magnet. And so what happens is the DNA fragments move towards the positive end of the gel. If you think about a magnet, right? You have a positive and a negative end. So how does this organize it? Well, the smaller fragments are going to move faster than the larger ones. And now once that happens, I've organized these fragments in a way that I can identify it again in the future. So now I have my fragments, and I have them organized by size. So now I have my tomato fragment, and maybe I have like an apple fragment, for example. I've organized all my fragments of a whole bunch of different fruits with specific traits. Now the fun begins. Now I get to interject them. So there's a couple ways we can do this. One of them is called recombinant DNA technology. This is when we take DNA from different sources and we can combine it to make new DNA. Specifically new DNA molecules. I blend up all these fragments together. And we call this recombinant DNA. So this is kind of the whole process of it. This is just an example here. But again, so I take my DNA. Let's imagine all of this is our DNA, the whole circle. I have found the part that I want to adjust. So I put my enzyme in there. It cuts it out. So I've taken my enzyme, I've cut it out, and then I'm going to use that gel stuff to organize it by lengths. Okay, this is the length I need, right? I need something of this length because this is the one I've taken out. I'm going to find the gene in there that does that. I'm going to inject my new part in there. And then I'm going to start breeding it, and boom, I now have my plant with a much better trait. So this is the process of it, right? But this picture is just this process here. So I've taken this DNA and I'm going to put it into bacteria cells, and then we study it. Why do we use bacteria? Because they breed so much and we can control them quite easily. So some ways we do this is through viruses or plasmids, which are, in bacteria and yeast. The thing that we use, though, as the glue is that DNA ligase. And you would have seen this word before. Ligase is that sticky stuff, right? That's what fills in those Okazaki fragments, for example. Don't really need to know that. So cool. Now I've inserted this into that bacteria. What the heck do I do with it? If I want to test this stuff, if I'm a scientist and I'm trying to really find the best stuff, I need a lot of this. And the easiest way we can do this is I just need to edit the genome of one thing. And then I place it in bacteria and it's going to multiply. It's a much better way of doing this. So if I need to make a lot of this, I'm going to mix it with bacteria and it's going to reproduce. So some of these bacteria take up the recombinant plasmid DNA through a process called transformation. So when we have this plasmid, they eventually end up replicating themselves, right? So we replicate this bacteria. And this is eventually what kind of leads us to clone it. So if you do this right, what you've done is you've cloned a bunch of bacteria with the DNA you specifically want. Now I have a bunch of identical copies. I can now start producing research on this stuff. I have a lot of stuff to play with. So we study these DNA frequencies with their nucleotides. Okay. So again, what the heck are nucleotides again? Right? It's these things, GATCs. So what I do is I label them. I fluorescently label them. It's a way to make sure you can track them, right? It's a dye. So what do I do with this? How do I study these sequences with this? Well, pretty simple. All I'm going to do is I'm going to take the entire genome, all the DNA, and I'm going to analyze it. I'm going to put it through a machine. It's going to sequence. It's going to tell me how much of everything do I have. So again, if we're looking at this, I have primed it. I put in my thing that cuts it out. I go through this gel electrophosphorus. It's going to take these things that I've tagged and it's going to sort them by length. If you've ever seen CSI and you've seen like a DNA thing on there or Dexter, this is what it looks like. This middle picture here, right? Or this picture on the right. And then I take this, this is my separated fragments, and then I put it in a machine and it's going to sequence it out. This whole thing is called PCR, polymerase chain reaction. Just so you know, whenever we reference PCR, it's that. Almost done. So we use biotechnology to fix things, right? There's a problem, we fix it. So when we have an organism and it has genes from another organism, we call it a transgenic organism. If I have a tomato plant, but I've injected some of the DNA from an apple into it to create a hardier fruit, it's a transgenic tomato. Sorry, transgenic. So transgenic animals, plants, and bacteria are used all the time in research, medicine, and agriculture. Those are the three primary things that we use it in. So we're going to go through these, just like what's the difference between these three? Animals are primarily used for research, right? We study diseases, we look at the food supply, we can look at health, and we can look at potential sources of organs for transplant. I'm pretty confident that someday there will be pigs that are bred specifically to be organ donors to humans. We already can donate pigs' organs. Pigs' hearts are a really common thing that's done as a heart transplant. So there is work underway right now to try and create more human-like hearts within pigs, for example, using biotechnology. Like, hey, there's a lot of people who need organs. There's a very short supply of organs out there. That is a big problem that this could help fix, for example. We look at transgenic plants. We usually do this for resistance against insects or pests. But there's other things. Sometimes we make it more resistant to weather. That's huge. Sometimes we try and make it bigger. We try and make it so that there's more offspring, more food. Maybe we want to reduce allergic reactions. Maybe we want to make it more nutritious. These are all examples there. And then finally, what the heck do we use bacteria for? Bacteria can be used to create insulin and growth hormones. We used to get insulin back in the day by murdering dogs. That's how early insulin was acquired. Now we can synthesize it. This is one of the ways how. We can look at, we can try and prevent ice on crops. We can use it to clean up oil spills. We can use it to decompose garbage. One big thing they're trying to do right now is to decompose plastic. That's one of the lead-in fields that people are using bacteria for now. They're trying to create something that can digest plastic. That would solve like immense pollution problem, for example. Okay. And so that is it for this. Keep that, sure. There is a third section. It's looking at the human genome. It's not that important. So it's one of the things I've kind of scrapped. So what's going to happen now is there's kind of three things that are happening. One is I do have the study guide for that. I do still want you guys to go into the textbook and glimpse through that. And the study guide is one of the ways you can do that. Use your notes as well. So there's three things you're doing today. One is the study guide. Then I also have two, there's a short quiz on just this topic, but three, remember that we had the genetics part A, this is the part B quiz of it as well. So this is 10, 11, 12, and 13. So that's what today is going to be. We're moving on from genetics after this. Today is the last day we're doing genetics. We're looking at genomes. We're trying to do a chapter or two a day, to be completely honest with you, because we still got two more sections to burn through. So again, three things: study guide, and there's the two quizzes that are on there to do. Oops, sorry.